Professor Werner Arber, welcome to be with us here in Lindau.
Thank you.
There are some years that have passed since you were receiving the big honor. At the time, what did you think would come out of being given the prize? Did it change your scientific work in any way or your general life?
Yes, well, when I got the prize, it's 25 years ago now. Of course, I pretended I shouldn't change my life. I did try to behave as before. I got, however, quite frequently obvious contacts from persons, from organizations, from groups, to ask me for giving some of my opinions, help in committees and so on. And I did realize rather rapidly that that was a good opportunity for me. I had the liberty to choose to say no or to say yes. And I think I made usually a good choice. Sometimes when I discovered that I shouldn't have said yes, I tried to get out of the business. And
give us an example. What was a good outcome after the prize, so to speak? Something
I have, for example, been asked in the 1980s. I was at the University of Basel, and I had been asked whether I would be willing to become rector of that university for 2 years' time. In fact, I would be a member of the rectorate during 6 years, because there was a rector designatis period of 2 years, then rector in full office, and past rector for another 2 years. So that was the group of the rectorate. After some reflection, I accepted, and it was really well seen, that I, with having been honored by the Nobel Prize, I wasn't thinking that I should do only science, and that brought me into science politics. And then sometime after, I had been asked to become a member of the Swiss Science Council, in which I have also been more than 10 years. And that brought me into really problems of science politics, which I think was good for my own country and also for the world altogether.
When you had your discovery back quite a number of years ago, there were many people
around
who said
1960, yes.
Yeah, there are. People said there are many risks. Can you see those risks today, or are people?
Okay. When I made the decisive discoveries in 1960, of course, it wasn't obvious at that moment what that would lead to. It needed to have the enzymes purified. That took another almost 10 years. And other people in our own lab contributed essentially to that. And it is then in the early 1970s that the enzymes which are able to cut the DNA into fragments, which allows to analyze the genetic information more properly than if you have the very long chromosomal filaments. At that moment, it became clear that certain fragments could be recombined differently together and also amplified in so-called gene vectors, which could be viruses or other small DNA molecules. That actually gave rise to genetic engineering, the recombinant DNA techniques. And at that moment, the question was raised whether there were conjectural risks into doing so. Now, I felt, because I had contributed to the knowledge that there are these enzymes and then the enzymes were used, I felt also a moral obligation to think about more deeply on these risks. And from today's point of view, I think I did contribute a lot for the evaluation of such more long-term risk, which would relate to the biological evolution. And I have studied for the last about 30 years intensively the molecular processes of molecular evolution. How do genetic variations, which is the basis for evolution, occur in nature? And are these similar or very different from what the genetic engineer can do in the laboratory? I work with bacteria, and I found out that there is a big opportunity to understand molecular processes by which the genetic information spontaneously undergoes changes. And this can be compared with what in genetic engineering is done reflectively by human beings. And perhaps I could just tell you that there are a relatively big number of natural processes on work which contribute each in its proper way to the formation of genetic variations at very low frequencies, of course, because for any living being, the genetic information must have a quite good stability. However, it's not absolute. If the stability would be absolute, there wouldn't be any evolution. And the evolution is a source of biodiversity. I think it's a wonderful thing.
But if you then go in and if you can control it, the humans can go on.
Yes, if you look at which kind of changes nature does spontaneously, there are in fact three major strategies. One strategy is within the genetic script changing one or a very few letters. These are local changes. The second strategy is within the genetic information of the organism, rearrange longer segments of that DNA, and sometimes duplicate one segment, or delete it, or invert it, or put it on another sector within the DNA. And the third large strategy is to acquire from another organism by horizontal gene transfer a segment which that receiving organism hadn't had before. These are the three major natural strategies of genetic variation. And their qualities, of course, with regard to contribution to evolution is different. Now, if one looks at what in genetic engineering is done, where also artificially, reflectively, DNA sequences are changed in order to make mutants on which one wants to study what is the effect of such changes on the life, on the phenotype. That can be locally, it can be by deleting a segment, by inserting another segment from another origin, and so on and so forth. So the strategies are just the same. And my conclusion for the time being is that we shouldn't worry too much about the risk of genetic engineering, because the risks are very similar to absolutely normal natural evolution, which is actually a natural phenomenon without which we wouldn't live today, and our surrounding world wouldn't be as it is.
Somebody said to me that the scientists who are dealing with basic scientists are different to those who maybe do medical research or develop certain kinds of products and so on with the technique, so that the moral high ground is high within the researchers and the scientists, so to speak, the basic scientists. But there might be other people who are not as morally. They might have other interests, financial interests.
Okay, if you're right. If a technique has proven to be applicable to something, of course, the inventor of that technique has no guarantee that someone else will misuse it for another purpose. That's clear. But this is true in any human doing. So that's a general problem, and it can be properly contained, I think, by bringing this particular issue into the political legislation, so that certain things are actually favored, and other things should be not allowed. That's a usual case. But of course, I do realize that this takes quite a number of years, going through all these political debates and so on, to have it finally implemented.
So it would be good from your point of view to have some kind of similar legislation all over the world on these issues.
Harmonization of the legislation in these fields would be very appropriate, and thanks to not at least through the activities of the scientists themselves. I have already recalled the Asilomar conference more than 25 years ago, have helped enormously to make all the countries attentive to these developments. And in many countries, the laws have been adapted to really conduct the appropriate research properly.
To be able to understand this valuable research that scientists are doing, and which often takes many, many years, you then have to be able to explain it as well. And I read a beautiful story that your daughter, I think, came up with. Could you just briefly relate that to us as a way of describing what you found out?
Well, Sylvia was 10 years old when I got the announcement of the prize in October of 1978. And the next weekend we went to some walks and we talked and what has happened during that week, which was really also impressing our two children. And then Sylvia said, if the other children in school tomorrow ask me what was that all about, what should I tell them? And then I tried to explain Sylvia in relatively simple terms what actually my contributions were. And after some reflections, he came up with some comparisons saying, well, DNA, is that correct? DNA is like the king, and these enzymes which you found, the restriction enzymes, are like some of the servants of that king. And I say, well, that's perfect. It's a good comparison. What is the role of the servants? And she said, well, if a foreign king, another DNA molecule, tries to penetrate into these cells, the servants kill that invading king by cutting it into fragments. And then I said, well, it's perfectly. And then I asked Sylvia, why don't you write this story up? And she wrote it. And I then included it into my CV for the book which the Nobel Foundation gives out. And therefore, later on, it has spread worldwide, this nice story.
Is it important to be able to, in simple terms, describe science today, natural science, to make it more popular and more understood?
I think it is important, but it also has some drawback. You know, one has to be careful enough so that it's not being misinterpreted. Simplifications help to understand better, but they also could lead to some misunderstanding. In this particular case of the king and his servants, I think there's no big problem. But if one uses simplifications too often, that can at long term be a source of misunderstanding of some principles of nature.
Some people have the feeling that there is now a mistrust between the general public and natural scientists. Have you got that feeling? And why is that in that case? Or is it rather mistrust of what one can do with science, so to speak?
It is, there is a certain mistrust. I think it has to do with human conservative opinions. Many humans are conservative for whatever. And if they don't see a major benefit for their own life, they are often very hesitant to accept technological progress. I think that's a normal human behavior. And some of these mistrusts, I would say, can, under certain circumstances, being misused by political movements, taking this on their flag. And I think good examples have been the peaceful use of atomic energy. And genetic engineering is another example, which has sometimes been misused in that sense.
For example, do you mean the debate around could we say genetically modified food or genetically modified species like, for example, dolly or?
It goes in this direction, without precisely what is behind. But I might add to this question, going further, after understanding how the molecular process of genetic variation works, me, as well as quite a number of other colleagues, have realized that these are, in fact, in most of the cases, done in nature by the help of particular enzymes. Enzymes are products of genes. And these enzymes often do nothing else than helping genetic variation to occur, and also, that's quite important, to not occur too frequently. So I say there are gene products which are generators of genetic variation, and some do it themselves, and some other gene products are modulators of the frequency of genetic variation to keep it low enough so that the species guarantees a certain genetic stability.
The positive uses, though, of your achievements are many. The way research and medical research have gone further. I mean, there have been a number of examples, for example, in the cancer research around cancer. Would you just give us an example that you are really feeling particularly proud of, where you know that your work has really benefited medicine?
Okay, well, if you go into the field of medicine, I think any very active protein like, let's say, interferons have been isolated thanks to these techniques. And then the question was, how should one apply them, in which kind of medical problems. There were some deceptions, but there were also discoveries that in certain other fields these products were very useful. And another example which has been widely successful is the product of human insulin, which made medicine largely independent of insulins harvested from living animals. And I think if one looks at these things, these are really very good examples. They're also in the human populations, in the society, relatively well accepted. While in other fields on which either people themselves or people in their families or in the neighborhood were not profiting from a treatment, people hesitate much more to accept genetic engineering and their products.
Were there times when you really despaired and said, no, I think I'm going to give this up? It's too much work, or too much, people might not believe in what I'm doing.
Well, I had periods on which, for long periods of time, my expectations were not fulfilled. I can remember that I had searched for recombinants giving a new type of restriction enzymes by either mutation or recombination. I worked on that for more than 1 year, quite intensively, without success. And I was very pleased only 2 or 3 years later that some other scientists working with a related system, but not with the one of ours, just found that spontaneously. We had been working on the ROG system. The principle can be found. I also know why in my system with which I worked, it would be much, much more difficult to find this kind of thing which I looked for. But that stimulates you, that indeed you're convinced that sometimes, even if your ideas might be wrong, you don't find the result. But that's how scientific life is. One shouldn't be disappointed.
So you need to be flexible. You need
Yeah,
to,
I think it is important, yeah.
and you need to share information
Yeah.
with other scientists as well.
Yes. The bad thing is, let's say if that happens to a very young scientist in his first experimental investigation, that's relatively bad for her or him. This, what I told you, occurred to me with, what was it, about 15 years of experience. And I was immune against reacting to that, I think, at that moment.
So sometimes it can just be luck as well. Hard work
Yes.
and luck.
Oh, yeah. Yes. And you have to have your eyes open. I was identifying the molecular basis of these restriction enzymes by working on a completely different project. That was in 1960, a time when the governments gave extra money to study the peaceful use of atomic energy, power plants. But some of the money had to be given for biosafety. And we did some experiments in such a project which was trying to find out the effect of different types of radiation on bacteria. And in doing that, they ran into the phenomenon of restriction, and within just a few months of additional experiments, I could show that the phenomenon has to do really properly with DNA. The modification is a methylation. And once I'd seen that, I felt, oh, this is an important discovery. You have to have your eyes open and take your liberty to do a few experiments to clarify what it is. And later on, I also had, of course, to tell to those people who guided the project that I had done something different than what I was supposed to do. But they understood that actually, after all, it wasn't so bad, and it was all right.
How do you relate that to young students of today? Your story and the way you worked, and what qualities do they have to go on?
Well, it needs, of course, interest, some degree of curiosity for how nature works. You have to overcome short periods of missuccess. And as I said before, you have to have your eyes open to the unexpected. Sometimes, each experiment that you do it in order to show something which is in your mind. But the experiments may be not giving that answer which you expected, but another answer. And if you just say the experiment failed and you discard it, you make never a new discovery. So, but it's not in all failure that a new discovery, of course, is inborn. So,
what are the big issues yet to be found out?
Oh, there are many. There are so many. Of course, we go more and more into trying to understand complexity, complex systems. And there is a lot of questions which even haven't been formulated yet, I think. There are more, how the proteins, which are gene products, mutually influence each other in their activities and so on. And also what I found in my own research, the coexistence within the genome of genes which are actually not working for your own personal life, for your individual life, but for the biological evolution of the population. I call these evolution genes. variation generators and modulators of the frequency of variation, which I mentioned already. And of course, the majority of the genes are other genes which are making your life possible, you know. I mean bacteria, plants, animals, and humans. And this is a very high philosophical content here, identifying that within the genome, there are not only genes whose product work for me, but contribute for further evolution, not only for human beings, but this is a general phenomenon, bacteria, all the living world, and gives the guarantee that nature itself takes care of the biological evolution, rather than that, as you still read in many textbooks, that evolution only benefits from errors and mistakes occurring to the DNA. Many, actually practically all of these genetic variations are actively introduced at very low frequencies into the genetic information. And of course, natural selection, that's Darwinism, will eventually decide whether such novel mutation is maintained or discarded.
But that is amazing. So those evolutionary genes, I mean, the way I could relate to it is that somehow we are connected, all of us.
That's correct, yes. Particularly since there is this third strategy of horizontal gene transfer. Whatever, somewhere in nature, novel genetic gadget has been developed, has some chance, at some later time, by chance, at low frequency, to be horizontally transferred to other organisms. So you can benefit from that, perhaps in some future time. So we are interconnected, of course, by common origins in the past, but we are also interconnected into the future by possibilities to profit from other developments made by others.
Which just makes it absolutely amazing.
Yes, it is. Yeah. So that is a kind, for me, you know, if you come to that kind of insight, if it touches you the first time, you think, well, it must be wrong now. Then you reflect. Then what is the consequence of it? There are a lot of philosophical, even theological consequences of that. You know, creation and evolution was often in conflict, you know. But what is going on in nature is, in fact, a permanent, slow process of creation, and that gives us a guarantee that also in future times, even if some living conditions are going to change, there is an internal natural potential that biodiversity will be amplified again. Of course, I have to add to that, without your question you may ask, this not necessarily is developing from humans to superhumans, but somewhere else in that evolutionary tree, there will be the branches growing.
You mentioned theology, as you said, there's often been a conflict. But
Yes.
theology has, I mean, a lot of people who are believers of any, different disciplines of religion always have this sort of saying, the connection between all human and, or actually all
Yes.
living beings in some of the religions.
Yes, that's right, yeah. Yes, actually, I think my understanding of molecular evolution is something which can give, from the side of molecular biology, a confirmation of these opinions of continuous creation. Also, I have talked to theologians, and it's an interesting aspect, you know. The question is, if you are believers on some religion like Christianism, the question comes up sooner or later, why does God, who actually likes creation, his work, and likes the organisms of creation, including the human beings, why does he allow that sometimes a genetic variation is against the positive life, genetic inheritance, inheritable diseases. And this has been widely discussed. But the duality of the genome, having genes working for your own, on the one hand, and working for evolution on the other hand, and we do know that not all genetic variations are beneficial. A majority, in fact, is detrimental. So genetic diseases are among those detrimental ones. So God, I believe, according to Genesis, found evolution as good, and that implies that mechanism that once in a while there's a sacrifice of an individual getting by chance a bad mutation. So that's how I think one could think about. I'm not sure whether that interpretation is correct, but it leads me into really considerations of this kind.
It's then, of course, I have to believe. Is it possible to be a believer and a scientist at the same time?
I think many of us can have both in parallel. We do not pretend that natural sciences can explain everything. There's other things behind.
Thank you so much for your time, Professor. I really enjoyed speaking to you.
You're welcome.
Thank you.
Okay.